We are on chapter 14, some carbohydrates. All right, so as we were talking about last time, carbohydrates obviously made up of carbons, hydrogens, and oxygens. There's really kind of two organic functional groups that we kind of see on carbohydrates. We do see again the aldehyde group on top of our carbohydrate. Remember again, we sort of refer to that as an aldose sort of group that has that aldehyde group. We also will have the ketone group present as well. And just like in organic chemistry, we also will call this a ketose because it has a ketone group on it. These groups are usually the furthest part of the carbohydrate. A lot of times they are put on top of the carbohydrate. And remember that we do have chirocarbons. So we can have some chirocarbons coming up here. And remember that a chirocarbon is basically a carbon that has four different groups attached to it. And the mirror image of that chiro sort of compound is non-superimposable. So that was kind of like your hand example. Again, if you put this hand up to the mirror, you get the other hand as the mirror image. If you pull out the mirror image, once again, with rotation, there's no way for it to fit perfectly over the original sort of structure. And that's a chiral molecule. It has a mirror image that's non-superimposable. And again, you can recognize a chirocarbon as a carbon that has four different groups attached. An achiral, on the other hand, has a mirror image that will superimpose, as opposed to a chiral where the mirror image will not superimpose. So kind of like a chiral compound, an achiral compound, or even an achiral carbon, we'll basically have not four groups that are the same, they're different. So we'll have at least a couple groups that are the same. Maybe it has two hydrogens that are the same. So for example, right here, this carbon right here, because it technically has whatever is above it, it has those two hydrogens that are the same. So that would be a achiral carbon. A chiral carbon would be, for example, something like this guy. And again, it has this, which is considered a group. That's a group that's different. That is different. And then pretty much it has everything above it, which is also sort of considered a group. So that would be, again, four different groups attached there. And that would be a chiral carbon. The one above it as well would also be chiral. And that one would be chiral as well in this particular case. So you can, as you can see here, and as we talked about last time, you can have obviously more than one chiral carbon within a carbohydrate. Now, when we talk about D and L, which we also talked about, that has to do with the OH group on the furthest chiral carbon from this group. So the furthest chiral carbon from either the aldehyde group or the ketone group. I would say, again, not 100% of the time, but pretty darn close. If you just back it up from that bottom guy, that's probably going to be a chiral carbon or the last chiral carbon. And again, not 100% of the time, but a lot of times it will be. And again, the location of where that OH group is on that last chiral carbon from the top there will give you whether it's D, which means it's on the right-hand side, or it's L, it's on the left-hand side as well. In terms of numbering carbons, we also do it very much like organic. So we want to give that sort of functional group the smallest number. So, that's always going to be carbon 1 up here, and then 2, 3, 4, 5. So this particular one that I drew, whatever it may be, it has 5 carbons. That's also known as a pentose, again, because it has 5 carbons as well. In terms of the ketone group, remember that the carbonyl carbon, because it has to be on sort of the second carbon in, thank you, uh, it will be on the second one in, so number one will be above it as well. There are monosaccharides, which obviously are individual sugar molecules. We can also kind of put this guy into a ring type structure and a cyclic sort of carbohydrate. And really, we get kind of two versions of rings that we could kind of use with it. One looks like a cyclohexane ring, but obviously is missing a carbon because there is an oxygen inside that ring, and it has only 5 carbons, which looks like so. And we also have a cyclic ring that looks like cyclopentane, but once again, missing a carbon because of the oxygen that's in the ring, has only 4 carbons. 1, 2, 3, and 4. So we talked about, how to kind of turn it into that sort of cyclic structure. Basically, you can just drop it on its side. And then when we do that, again, everything on sort of the right-hand side there will end up down in terms of their groups, and everybody on the left-hand side there will end up. And that's sort of how you can put those locations. Remember, kind of to the left of the oxygen on each of these is really where that last sort of carbon will end up, and it will end up there above the ring. So that would be like that guy right there would end up above the ring. Usually, the fifth carbon on a hexose, which has 6 carbon chain, is the one that goes and attacks. But usually, the last chirocarbon there will go and attack wherever the carbon oxygen double bond is. So, if it does have the aldehyde group on the top there, that's going to go to the top carbon, number one. If it is more of a ketone group on that carbohydrate, it's going to go to the second carbon, most likely is where you're going to find that carbon oxygen double bond. Remember, you could kind of count how many carbons will be in your ring and sort of start with your ring. Remember that where it comes together, for example, like on the fifth and the first carbon, where it comes together, we kind of skip that. And we end with the last sort of OH group on those carbons over here as either being alpha, which basically means it's going to go down, or beta, which means it will go up. So if you're asked to draw either the alpha or beta, that's really the position of the last OH group there on that carbon ring on the right. Any questions on any of that stuff there? All right, so here's the one again that we maybe went through, but again, you can number from starting with 1 and coming all the way down. Once again, you can basically just dump it on its side. And as you can see here, and as we talked about again, everybody on the right there basically will end up down, and everybody on the left will end up up. So all these guys originally were on the left-hand side, and these guys were all on the right-hand side. And in this particular case, it is going to be that fifth carbon that's going to attack that. And that sort of area right there is really what's going to tell you again what ring you should draw. You could literally just circle the carbons. There's 1, 2, 3, 4, and 5 carbons. And there's 5 carbons that are going to be involved when it comes together as a ring type structure. And that means you would draw the one that kind of looks like cyclohexane is sort of the ring that you would want. And you would start with that ring right there, which again is our 5 carbon ring. Again, number 6 going to the top. And then we come down to the ring and we do number counterclockwise. Again, starting with this as obviously number 6 up here and coming down 5, 4, 3, 2, 1. Again, skipping the fifth as it gets incorporated over here and going to our fourth. And again, if we just back it up there and take a look at it, as we can see, number 4, the OH is down. Number 3, the OH is up. And number 2, it is down. So it's kind of down, up, down in terms of what we got going on there. And that's what we see here, down, up, and then down. Once again, this last one is determined as either being alpha or beta. You're usually asked again to draw one or the other version of it. And as we just talked about there, going down on that last one there will be alpha. and going upwards there will be beta in that particular case. Now, remember, I think we did want the very end there where it is possible, especially when you have that sort of ketone group present, that you actually will have maybe the smaller ring and you'll have two carbons that are not part of the ring structure and sort of off of the ring when you count it up. Any questions on that there? All right, so again, just to kind of show you how sort of the oxygen gets incorporated, here is our carbon number 5 with our OH group. And here's our carbon number 1 with our aldehyde group. And again, as you can see, as they come together, that hydrogen actually kind of jumps over here. And that oxygen that was there is how it gets sort of incorporated into the ring at that point, which is why we have an oxygen sort of in that position. And then that OH kind of works its way over there to that double bond and breaks a double bond and makes basically an OH group there at number 1. So again, because that kind of where the ring comes together incorporates that oxygen, that's why we skip that one when we're kind of going up and down on our ring because it just gets kind of in there. And we do end up with a new chiral carbon here. Once again, it has one group, two groups, and really 3 groups and 4 groups happening up over here. All right, let's take a look here. Let's try this one, man. So why don't you try, let's do one here one more time. Let's go. Uh, uh, and again, what we do, let's do the beta anomer. All right, so take a few moments and draw that guy up there and see what you come up with. Bless you. Carbons, so 1, 2, 3, 4, 5, and 6. Again here, we're going to go from carbon number 5 in this case of our hexose, and it is going to go and find our carbon oxygen double bond. And we can count up the ring that we need to draw by just doing that. So that is 1, 2, 3, 4, and a 5-member ring. So that's the one again is going to look like cyclohexane, but with the oxygen obviously incorporated inside. I'm not going to redraw it on its side. I could just remember that if it is on the right, when I drop it on its side, it's going to go down. And if it's on the left here, it's going to go up. So again, if you don't want to redraw, you can just kind of remember that and it should work out all right. We do know that number 6 here, which is not part of the ring, does always end up here to the left here of our oxygen. And again, that gives us a point of numbering. And we're going to go counterclockwise when we hit the ring. So that's going to be 5, 4, 3, 2, 1. So as we saw in that previous slide, that OH group there on carbon number 5 gets incorporated into the ring, which is where that oxygen comes from. So we are going to skip number 5. And we're going to start with number 4 here. My OH is on the right, which means it should go down. Number 3 here, my OH is on the left, which means it should go up. OH on the right-hand side here for number 2, which means it should go down. And lastly here, that is what's going to determine where the last one's going to go. And beta pretty much means up, so we're going to pop it up there on the top. Any questions on that one there? Yeah. You do not. A lot of times they will be left off again, as we talked about last time. Sometimes it gets a little crowded if you kind of put the hydrogens. But you are correct. And just a reminder that on the other side of that line, there are hydrogens. You can. It's not necessarily wrong if you want to put it there. But sometimes the book and stuff will kind of leave it off just because it gets a little crowded around with all the hydrogens on there. Yeah. But yeah, that's a good point. It's not. It's definitely not a carbon like an organic. That's going to be a hydrogen on the other side of that line. Other questions? Okay. All right. Now, alpha D glucose and beta D glucose in solution sort of will open up and close. And alpha D glucose converts to beta D glucose and back. There's only a small amount of the open chain that's there. You can actually see in most of the case there, 64% of the time, it's hanging out there in the beta position, once again, with our OH group hanging up there on the top, as opposed to our alpha where it is hanging there on the bottom. And once again, basically as this closes, that's going to go there and he's going to jump off and that's going to become a single bond. And again, going to pop over there as it kind of closes in on itself. A lot more takes place than just moving over there and drawing it, but that's essentially how where everything sort of ends up as you close it down. We did one similar to this, and again, when we look at something like fructose, here we are going to look at our carbons, and this would be carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Here at carbon number 5, it's going to actually once again go to carbon number 2 in this case, because that is where the carbon-oxygen double bond's located, and it happens to be at carbon number 2. Once again, because of that, we do get the smaller ring. So we got 1, 2, 3, and only 4 carbons that are going to be part of the ring structure that you're going to build. And that is again why we draw this guy here, 1, 2, 3, and 4 carbons. In this case, we actually have not one, but 2 groups that are hanging off of it. And we still start with this one as carbon number 6 in this case. This would be 5, this would be 4, this would be 3, this would be 2. And that carbon number 1 that's also off of the ring actually ends up over here. And again, as sort of a group, depending on which one you're drawing, either the alpha or the beta, as to where that sort of carbon group at number 1 will end up. If you're doing the alpha, you got to put the OH there, and that's going to put the carbon group going in the upwards direction. If you're doing the beta, it's going to put the OH up and drop the carbon group to the bottom. So when that's off of the ring, sort of where that carbon group ends up is sort of determined on, is it alpha or beta with the OH group that is there. Any questions on that one there? All right, so maybe instead of this, let's talk about, since we've done a number of examples of this, let's talk about if we did have one, say like, let's just go with, I don't know, do something like that maybe. And let's do that, and let's do that, let's do that, and let's do this. All right, so we got this sort of cyclic carbohydrate. By the way, it's alpha or beta, this one is alpha, right? Again, that guy heading downward, right? So it's alpha. All right, let's just say we want to like open this thing up, right? And we want to open it up back into its sort of non-cyclic form. We can do this, and we could do this by just looking at sort of what we have going on. And let's just say we wanted to open it up into its D form here, right? So the first thing we should notice is when we look at this ring, that again is the cyclohexane ring, less 1 carbon, which means on the ring, I got 5 carbons. And I got 1 carbon off of the ring, which would mean that I definitely know that my carbohydrate has 6 carbons in this case. Because of that, I got 5 on the ring plus the 1 off of the ring. Gives me a grand total of 6, right? So I know that the carbohydrate in its open form is a 6 carbon carbohydrate. I also know that that's going to be 6 up there on top, which means if I do my numbering, that's going to be 5, 4, 3, 2, and 1, obviously going like we would put it on there. So at that point, I could actually start here. And I also see that if I look at this ring, I only see sort of one carbon group that is not part of the ring. And usually that means that that probably came from a aldehyde group, right? As we saw when we had our ketone group, where we just looked at fructose, we ended up with a couple carbon groups off of the ring, one at kind of six and one at the front end there. And in this case, we don't have that. So you could probably feel pretty safe that this carbohydrate has the aldehyde group up on top. Otherwise, again, you would probably see some type of carbon chain hanging out over there on the other end, and it probably would be the other ring structure that you probably would have as well. So with that, I got one carbon there. We'll go two carbons, three carbons, four carbons. We'll go five carbons, and we know that carbon number six there is going to be off the ring there. So this is one, two, three, four, and five. And that would be number six right there. All right, so I have really the basis of my structure here. Now, just like we would put it on the ring, left is up, right, when I drop it on the side, and right is down. We could kind of go backwards the same way, basically. So we'll go to number four, and I see my OH group is down, which means when it's on its open chain, it should be on the right-hand side there, right? And obviously I would have an H on the other side. If I come to number three, it also is down, which means it should end up on the right-hand side. Number two there is up, which meant it probably started on the left-hand side for my OH. And now the only one I have left is number five, and in this case I want to do the D, so it should be on the right? So that's going to come from here. So kind of like alpha or beta gives you kind of where you put that last OH, up or down. Obviously, if you're going in the opposite way, the D or L will tell you where to put that OH group on that last chiral carbon. And obviously, we would have an H over here. Any questions on how to go backwards and open it back up? Yeah. All right, so why don't you try one and see? Maybe. Actually, I'll do it here. We'll do it here. So let's say we had uh, uh, so why don't you draw we'll do the L form there. So open that up and draw the open chain from that one. Okay, so let's take a look at, so once again, a good place to start if you're going to open this up is just to figure out how many carbons you got going on. Once again, when you see this ring, that does represent there's four carbons obviously within the ring. One, two, three, and four. Now, outside of the ring, that is five carbons and one more makes six. So once again, here we do have a six carbon carbohydrate here. I also see a couple of things that we just talked about. I see a couple of carbon groups hanging out there. Probably could only occur if you got a ketone group happening there. So just like in organic chemistry, right? One, two, three, four, five, six. You gotta be second in for that ketone group, right? So it cannot be at the end. That would mean that this again is going to be carbon number six. This is going to be five, four, three, two, and now number one is going to be off of the ring, which makes it pretty simple there for this guy here. That's going to be carbon number one, number two, number three, number four, number five, and number six. Again, that ketone group does need to have the smallest number, so obviously that is the way we're going with that. So we also know where number six is going to be as well. So there's number six as well. So all we have left now is just kind of the little pieces there. So number four, my OH group is down. And again, when you drop it on its side, everybody on the right-hand side will end up down. So number 4 there should have an OH group there. Number 3 also down, which means OH group will be on the right. And in this case, they do want you to do L, which means we will throw that last OH group there on the left in this case. Any questions on how to open it there? All right. Question on to draw it into a ring, which you might need to be able to do on the final, perhaps. Yes. So make sure you know how to do that, perhaps. Uh, or opening it. Yeah. Okay. All right. Let us continue on there for sure. All right. Let's talk a little about some properties of monosaccharides. Monosaccharides are reducing sugars. And these are monosaccharides with a carbonyl group that oxidizes to give a carboxylic acid. That would react with Benedict's reagent like you used previously as well. And remember, we used Benedict's reagents on that aldehyde to carboxylic acid oxidation that occurs, right? And again, adding the oxygen in there. It includes pretty much all monosaccharides, glucose, galactose, and fructose. And here you can see that pretty much same situation as we talked about really in organic chemistry. We're really doing the oxidation step here. We're going to do the definition of oxidation of adding oxygen in, and that is going to give us our carboxylic acid group, just like it did in organic chemistry. And remember that we will see that happening, and that will reduce down our copper in this case, from copper 2 to copper 1, which takes it from a bluish color to that orangey reddish type of positive color that you get with the Benedict's reagent as it's being reduced. The reduction of monosaccharides involves a carbonyl group that actually produce what are known as sugar alcohols, such as D-glucose gives us a sugar alcohol known as sorbitol. You don't have to worry too much about that, I don't think. All right. You don't have to worry too much about this, but really, again, what is the result of the oxidation of this? Oxidation is going to occur pretty much at the organic group. And again, that's an aldehyde getting oxidized, basically to a carboxylic acid. So all that's going to happen is we're just going to pop an H in there. Everybody else is going to be the same. 2, 3, 4, 5. Not much else is going to change here other than that move right there of just adding in that oxygen to get us that carboxylic acid. So that's basically the only thing that's going to happen in this particular case. And just like pretty much organic chemistry, this is your primary alcohol to your aldehyde to your carboxylic acid. So that's pretty much the exact same step along the way. And obviously the reversal of this is all oxidation basically occurring. And reduction going in the opposite direction, right? As you go that way to that way. So once again, you can see nothing below the functional group pretty much changes. Everything's the same. Just that functional group is basically what happens. All right, let's then talk about a little disaccharides. So disaccharides consist basically of 2 monosaccharides that have been put together. Pretty much you just take out water and then they kind of stick themselves together and they bond to each other. So you will need to know these. We're going to look at each of these individually as we go through it, but it's a good slide. It kind of lays them all out there. So the disaccharide maltose is made up of 2 glucose molecules. Lactose is made up of glucose and galactose, and sucrose, which is sugar there, is made up of glucose and fructose. So let's take a look at each of these individually here, but you do need to know what maltose is made up of, what 2 monosaccharides, galactose and sucrose, what are the monosaccharides to make those guys up. So maltose is known as malt sugar, is composed of two D-glucose molecules, and they are linked together by a special type of bond, which is known as the 1,4 glycosidic bond, which is a fancy way of saying where the things are bonded together, what carbons are involved. And we'll take a look at it right there in just a second. Used in cereal and candies and also in brewing as well. It is found both in the alpha and beta form. So here are two D-glucose molecules. And basically what's going to happen is right here at carbon number 1 and carbon number 4, hence part of the name there, the 1 and the 4 of what carbons are involved, are going to come together however you want to think about it, but essentially you're going to take out water. You're going to take out water and they're going to be joined by that oxygen. And that gives you what is known as an alpha 1,4 glycosidic bond, which for the most part kind of looks like what's in the box. And what I mean by that is we learned earlier alpha means down, right? So if you kind of look at it, the lines are kind of coming down to the oxygen. So that is what's sort of happening there. And that's sort of what that alpha 1,4 bond is happening. It is only alpha because that is alpha there. That is not alpha. Alpha is over here on that carbon. So there's no two alpha or betas involved. So it's just one alpha. So you need to know that maltose is made up of two D-glucose molecules held together by that bond, the alpha 1,4 glycosidic bond. Yeah. Any questions on that there? Made obviously by removing water, which means if I wanted to break this guy back apart, I throw some water back in and it will break back apart and give you both of those carbohydrates obviously back together. Now, lactose, on the other hand, is made up of a beta D galactose, spit that up, beta D galactose, and an alpha or beta D glucose molecule coming together. And it contains a slightly different looking bond or named bond. And this bond is known as beta 1,4 glycosidic bond. It is again the 1 and the 4 are the two carbons that are involved. So those are the two carbons that are coming together. And same deal here. We're just going to take out, however you want to think about it, doesn't really matter for our purposes. We're just going to take out water. So two hydrogens, obviously, and an oxygen. That still leaves us one oxygen, which is sort of how they bond together. And that creates this beta 1,4 glycosidic bond. And a good way to remember that is it kind of looks like this, like it's kind of going upwards, and beta means up, right? So if you see this bond that kind of looks like it's going upwards, that's going to be a beta 1,4 glycosidic bond if it involves those carbons, as opposed to the one we just saw in the previous one, which kind of looks like it's going downward here, and that's going to be alpha on the previous one. So again, those are kind of a good way to kind of maybe visually sort of remember those things with the alpha and the beta kind of going upwards in this particular case. Obviously, lactose is found in milk and milk products, and sometimes people have a hard time digesting that and breaking those bonds there in the body. Lastly, here we have sucrose, which is table sugar. It is made from a alpha D glucose and a beta D fructose. And I believe this picture is incorrect. That OH, I think, should actually be going downward there. Here we go. And in this particular case, we have a different connection. And the connection actually is between carbon number 1 and carbon number 2 over here. And that is right here where my alpha D glucose is hanging out there. So that's kind of in the way there. Let's see here. I'm going to get rid of that there. Can I get that out of the way? Maybe. There we go. Now you can see it. All right, now you can actually see it. There we go. All right, so carbon number 1, and again, that should be down that way, I think. Carbon number 1 and carbon number 2 on the fructose. So that's going to give us a 1,2. This is a alpha beta 1,2 bond because it is the carbons on the right-hand side of the ring, which is where we find alpha or beta. And since that is the alpha carbon there from the glucose and the beta carbon from the fructose, we use both of the alpha and beta. On the previous ones, we were using just the alpha and just a regular carbon, or the beta and the regular carbon number 4, so we didn't have both the symbols there. This is an alpha beta 1,4, also made by removing the water. Basically, the H2O comes out between the two, and we put those guys in here, and we get our bond that's happening in this case. This obviously is table sugar there, and this is also one that cannot be reduced. It's not a reducing sugar because all that alpha and beta section of those guys are in some trouble because they're being used in this bond. Yes, sir. Honestly, I would ask that on the exam. Uh, Two types of questions, yeah. So, where two monosaccharides to make up sucrose, and you would not be asked to draw it, but you may be given the picture and maybe asked what type of glycosidic bond you're looking at. Yeah. I would say for these three, the maltose, the lactose, and the sucrose, you should know the glycosidic bond that's associated with that, those guys coming in. But just in general, you may have a picture that you may have to recognize, or identify what type of glycosidic bond. You won't be asked to draw it, but it may be provided the picture for you. Yeah. Other questions? Yeah. It is in this case because it's the alpha carbon from the glucose coming together with that beta carbon. And again, we didn't use both of those sort of symbols, if you will, say on this one, because this was the only sort of alpha or beta carbon that was involved because we're doing this one and this one. This is where the alpha carbon is on that guy, and it's not involved in the bond. So that's why that one was just called like alpha 1,4. And just like on this one, this was the beta bond involved. But again, that's the alpha bond over here. And this is not an alpha or beta bond on that carbon. So that's why, again, we just use the beta. In this particular case, we're putting together over here. This guy, if we just drew sort of this part of it, we had the alpha sort of carbon involved for the top guy. And for the bottom guy, we had the beta guy happening there. And then on their side here, obviously, is the CH2OH. So in this case, we're actually using both the alpha and the beta coming together to make that bond, which is why it's in the name for that type of bond that's happening in that case. Yeah. Other questions? All right, so to answer your question again, you need to know the monosaccharides to make it up. You need to know specifically those glycosidic bonds for those three things for sure. Yeah. All right, you're not going to be asked, obviously, to draw any of this, but as we were talking about, just in general, if you do have a disaccharide, obviously, we took water out to put them together. You would put water back in to break them apart. And obviously, for example, here, it would be broken apart here, and that should still be down in this direction, I think. And obviously, you would just really give each one an OH group, right, where you break it apart. Nothing else will change. And obviously, if it's the alpha glucose, like it is in this case, you put that OH down. And on that guy on the bottom there, that's going to come from fructose, which is beta. You would put the OH coming up on that case. As you can see here, hey, they got the OH on that side, right? That's good. So there's our alpha, and then there's our beta. I don't worry about sweeteners. All right, so for each of these here, take a moment and decide which monosaccharides are involved in these disaccharides. Like a question you might find. Okay, so lactose, as we just saw, should be perhaps a glucose and a galactose happening. Maltose was the first one that we looked at, which is a couple of, let's circle it twice, how's that? Two D-glucoses coming together. And then sucrose was the one we obviously were looking at the last there, where we had our alpha beta 1,2 bond. Again, glucose and fructose in this case. Maltose again with the alpha 1,4 glycosidic bond, and lactose doing the beta 1,4 glycosidic bond. That's happening there. Any questions on those there? All right, we're gonna take a 5-minute break and we'll wrap up the rest of this chapter. Yeah. So we've talked about glucose here, carbohydrates, and disaccharides. We're then gonna finish up here talking about polysaccharides. So disaccharides are basically putting together a couple of monosaccharides together, basically removing water to do so. Polysaccharides are pretty much just putting a bunch of them together, removing waters along the way. So they are long chain carbohydrates that are, like I said, basically put together through glycosidic bonds where we remove water. And they just kind of bond together. Some are straight chain, some are branch chain, like we see in organic chemistry. So let's talk a little bit about polysaccharides. Again, they are polymers of D-glucose. And in this case, they include things like amylose and amylopectin. They are starches that are made up of alpha D-glucose. They also include glycogen, which is animal starch in the muscle. We do save some of our glucose through glycolysis as glycogen that we can use through gluconeogenesis to break back down to use for some energy. They also include things like cellulose, which are plants and wood, which is made up of beta D-glucose in this case. So basically, a lot of it is just a bunch of these guys put together, again, linked through those glycosidic bonds from the remover of water. So let's talk a little bit about the structures of amylose and amylopectin from our baked potatoes, perhaps. And here we have amylose, which again, these are all really glucose molecules. They're all put together. And once again, in this is a straight unbranched chain. So it's just a long straight chain of these guys together. So like in organic chemistry, just like a main chain, nothing coming off of it like branches. Once again, they're all put together through the alpha 1,4 glycosidic bond, just like we were talking about with our disaccharides. That is from our alpha carbon there on our glucose to carbon number 4 here on the other glucose molecules. So once again, that's the alpha 1,4 glycosidic bond, just like we saw with maltose. Kind of looks like it's coming downward, right? So alpha is a good way to remember that. Again, made by removing water basically to do so. Amylopectin is pretty much the same deal. The difference, though, is it is not an unbranched chain. So there's some branches that come off of it, kind of like an organic chemistry where we have kind of a main chain and then we have things coming off of it. So we do have the exact same, if you will, kind of main chain that we see with amylose there. We have our D-glucoses held together by our alpha 1,4 bond, as we see here. But every so often, we start to get some branching that's occurring here between the glucose molecules. And this is going to give us a different type of bond that's happening here. And this is a 1 bond, which is an alpha carbon off of that glucose, which originally looked something like that. And this is actually carbon number 6. This guy right here was our CH2OH group that we put kind of above the ring there coming off. And that is where the numbers come from. Carbon number 1 here and carbon number 6 coming together. Once again, we're going to remove water as a result of this. And you can see basically the OH here and one of the H's removing our water. And that's going to give us our alpha 1,6 glycosidic bond. So here we have 2 different glycosidic bonds happening, the same one that we see in both for the main part of it, but on the branching points here, they're branching between carbon number 1 and carbon number 6 there between our different glucose molecules. So amylose is again unbranched, held together by the alpha 1,6 bond, while amylopectin, again, going to have a little bit of branching that's happening, giving us two different types of glycosidic bonds, alpha 1,4 and alpha 1,6 in this case. And again, to an earlier question, maybe you have a picture like this, you need to identify kind of that bond that's happening there between the carbons. Glycogen is a polysaccharide that stores glucose in muscles, like I said, for later use in case you need to kind of burn that through gluconeogenesis. Is similar to amylopectin, but it's a lot more branching going on. So similar bonds that you have in amylopectin, your alpha 1,4 and your alpha 1,6 bond that's happening at the branching points, but just a lot more branching happening. And again, because it's basically storing all those things, so they're trying to get everybody in there together as much as they can for storage. Now, cellulose, on the other hand, is the polysaccharide of glucose in an unbranched chain. It has a different one. It has that beta 1,4 glycosidic bond, again, similar looking, hopefully, to the previous one that we saw. Again, kind of going in the upwards direction. Carbon number 1 of this guy, carbon number 4 of this guy. And once again, kind of like that oxygen's going upwards, just like you find in beta. And again, that's a result really because this guy was originally a beta OH group that's happening there. So it's coming upwards in this case. So also still our D-glucoses just held together by that beta 1,4 glycosidic bond. Cannot be digested by humans, which is probably why you shouldn't eat it, I imagine. You cannot break that beta 1,4 glycosidic bond. All right, so take a moment for each of these. Identify cellulose, which has the beta 1,4 bond. And again, that is the one that sort of looks like it's going upwards between the glucose molecules. Here we have two things. We have the amylose and the amylopectin. And the amylose, we have the alpha 1,4 bond, which looks like kind of going downward. The amylopectin also has the alpha 1,4 bond, but it also has the branching that's occurring. And again, that is going to give us the alpha 1,6 bond coming downward from the alpha carbon here to actually our 6 carbon on the other guy coming down that way. So again, we got the alpha 1,6. And obviously here, that's glycogen, which is basically the same bonds that we just talked about, but a lot more kind of happening, a lot more branching sort of happening in that situation. Any questions on any of those things there? All right, that wraps up chapter 13 of carbohydrates, they say. 13, I think. 13? 13. Yeah. Yeah, I don't know why like most books keep all the organic chapters together, but she decided, I don't know, in this version, like, hey, we'll just throw carbohydrates before the last organic chapters. All right, so in this chapter, we're going to talk about proteins and amino acids. And we're not going to cover everything in this chapter, so we're just going to cover certain things, and you'll be responsible for as well. So let's talk a little about function of proteins. Proteins obviously are made up of amino acids, and there's different types of proteins. There's structural proteins. There's transport proteins, which carry essential substances throughout the body. We also have hormones, enzymes, protective type of proteins as well. Now, pretty much all proteins are made up of amino acids, and amino acids, that's good, they messed up too much, are really the building blocks of proteins. Different sort of combinations of amino acids come together, code for different proteins along the way. But all amino acids pretty much have a similar structure, and you should be familiar with the structure here. And basically, it has, if you want to think about it like a organic chemistry type of situation. We have a carbon. It has a carboxylic acid type of group. It has really an amine type of group or an ammonia type of group, and it has a hydrogen. And it will have this R group, which is sometimes referred to as the side group. And it will be different for different amino acid. There's like 20 of them. And what we'll see here is kind of two versions of this. A lot of times it is drawn as the ionized version, which is sometimes referred to as the Zitteron ion. And that is what we see over here, where our carboxylic acid has lost the hydrogen and becomes negative, and basically our NH2 gains a hydrogen and becomes positive. This right here, just so we are clear, is still the carboxylic acid group, but missing a hydrogen. So that is what's going on over there. And obviously this is basically ammonia or an amine. And then we obviously have a hydrogen. So I would say most of the time you'll see this sort of ionized version of the amino acids. You do need to know the sort of structure of an amino acid in a sense. What are the different parts of it? So again, kind of like a carboxylic acid part, an ammonia or amine part, a hydrogen, and some type of, most of them are carbon containing side groups, except for one, which is just hydrogen. But the other ones will be usually some type of carbon containing side groups. Here's an example of a couple of amino acids. This is glycine. Its side group is our hydrogen. Here, alanine is basically a methyl side group in this particular case. But again, you can see in terms of them, they all have the similar structure: carboxylic acid, NH3 plus, hydrogen, and carbon on each of them. So that is universal for all the amino acids. It's also important for how they come together and make a peptide bond, which is really the same bond that we see in organic chemistry, or we saw earlier as well. All right. So you will not need to know the names of the amino acids. You will need to know the general structure of an amino acid, and you will need to classify an amino acid as one of these four types, as either being a nonpolar, a polar, an acidic, or a basic amino acid. So you won't be asked to draw like glycine, or won't be asked to draw alanine or anything like that, but you may be given the structure of an amino acid and asked to classify them as one of these four types of amino acids: nonpolar, polar, acidic, or basic. So we're going to kind of look at the differences between these to help you sort of identify sort of what category to put them in if you see the structure. So we'll start with nonpolar. So nonpolar, it's just like we talked about all through chemistry here this semester. It basically has equal sharing of electrons. It's basically neutral. As we know, because of that, they use dispersion forces, which means they are typically hydrophobic, which means they don't like water. So hydrophobic, right, means water hating, basically. So they typically will have a carbon hydrogen type chain, kind of your basic carbons and hydrogens, nonpolar group on it. So if you see an amino acid that's basically just that kind of carbons and hydrogens, not much else going on that side group, that's going to fall under the nonpolar category of an amino acid. Polar means that it has some type of polar group, which, just like we talked about with bonding, means that there's going to be unequal sharing of electrons. And because of that, there's going to be like a negative side to a bond or a positive side to a bond, which allows it, for example, to do something like hydrogen bonding, right? Like we talked about previously. So those things are typically hydrophilic, which means they like water. They're water loving. So don't like water, right? Likes water, right? So these typically will have some type of polar side chain. So in addition to carbons and hydrogens, you'll see something like an OH group hanging out there. And that OH group, right, is the same sort of OH group we see in water. It can do some hydrogen bonding. and that's going to be in the sort of polar category of amino acids. Acidic amino acids also are going to be hydrophilic, which likes water. They typically will have an acid sort of side chain. This looks like, hopefully to you, a carboxylic acid. So that's a great way to remember that, right? Looks like a carboxylic acid. I should probably put that in the acidic sort of category of amino acids. Lastly, basic ones, which are also hydrophilic. They like water as well, and they will typically have some type of nitrogen side group there. A nitrogen that's attached to a hydrogen, like an amine type group or an amide type group. And these are basic ones, right? So a good way to remember that, hopefully, is NH3 is a weak base. So if you see anything that kind of looks like NH3 hanging out there, or some type of derivative of NH3, is probably going to go into the basic category. All three of these guys like water because right there, you could hydrogen bond, no problem. Right here could use some hydrogen bonding. And obviously, that's a nitrogen attached to a hydrogen, which means it could also hydrogen bond just like ammonia could hydrogen bond. So they have those sort of groups that can participate in hydrogen bonding with water, which makes them water loving. Again, here, nothing much going on there, but a non-polar sort of group. So once again, going to have to rely on dispersion forces, not going to work well with water. So here is some pictures similar to what you'll find in your book and our classification. So these are all non-polar amino acids, and for the most part, most of them are just carbons and hydrogens. We do have sort of a nitrogen over here, but that is just a lot of carbons and hydrogen. If you remember from our conversations about in organic chemistry, what happens to sort of interaction with water and solubility with water. Once you have a lot of carbons, that goes out the window. So even though it does have a nitrogen on that guy, that's a lot of carbons in the way, right? And so that's going to have some steric hindrance, and it's not going to be able to interact well. So again, lots of carbons here, your hallmark of sort of your non-polar amino acids. Here are some examples of polar amino acids, and once again, we do see several sort of common threads here. We definitely see some OH groups hanging out, so that's a really good sort of idea. Remember that SH, like we see in a thiol group, right, is similar to OH. So that's really good as well. And here we have this group here, which is an amide group happening, right? So that is our amide, right? Our carbon oxygen double bond with the amine at the end of it there. And again, these are really just the same functional groups we saw in organic chemistry, right? So we basically have anything with an alcohol group on it, a thiol group, or an amide will go into our polar amino acid sort of category along the way. Acid and bases here, again, anything with like a carboxylic acid looking group, going to go into the acid. And as I mentioned before here, we have a lots of kind of NH groups hanging out there. So those guys are going to go into our basic group. So something that looks like NH3 will be there. Any questions on how to classify them there? So again, you're responsible for the final basic structure of an amino acid, being able to, if you're provided some pictures of some amino acids, being able to put them into those sort of categories. Yeah. Any questions on that there? All right, so take a moment for each of these here and classify what group maybe they should be. Should they be polar or non-polar happening there? So let's take a look. So the first one might be hard to see because the side group is actually a hydrogen. So that is the side group there. And again, we have our basic sort of amino acid structure happening here. So in this case, that's obviously a hydrogen. So that's going to be non-polar. Over here, we do see an OH group, right? So that tells us something like water, gonna like water. So something that's gonna definitely fall into the polar category in this case. Any questions on those two? Now, Fisher projections of amino acids, they are chiral except for glycine. So glycine is the one with the hydrogen side group, but all other amino acids will be chiral at that carbon because they will have four different groups attached. Obviously, glycine, which is the one we just saw, has one and two hydrogens attached. So obviously not four different groups attached to it, along with obviously our. So it's got not four different groups. So these guys will have stereoisomers, which means that there will be kind of a D or L version of each of them, kind of like what we did with the OH group there on carbohydrates. Again, L meaning left, D meaning right. So in this case, we're looking at our NH2 group, our amine ammonia type group. And here that is on the left, so that's going to be L. And here that is on the right. And that obviously in the middle there is our carbon that is there. So here again, we see L and we see D that's happening here. Now, the Zitteron ion is the ionized version of it. So sometimes people will talk about amino acids as the Zitteron version, and that's just a fancy way of saying it's basically ionized on both sides there. So again, we won't have the H on this side, but we will have an extra H on that side, and that'll be our ionized version of it. Basically, that happens. So if you see Zitteron, it basically just means you're looking at the ionized version of it, which typically happens in solutions and so forth. They will get sort of ionized based on the water being present. All right. We don't care about isoelectric points because I can see it. See, you don't care. Not sure I care about it as an acid or a base. Not sure I care about the pH of it. I'm pretty sure I don't care about electrophoresis, which, by the way, is to separate it out of everybody through a gel by size. You can put some X's on it as I go through it. I'll let you know when you should stop Xing things out. I don't care. Still not caring too much. All right, I'm going to start carrying again here. Right about here, I'm going to start carrying. All right. So let's talk a little about the formation of peptide bonds. And peptide bonds are the bonds that basically hold two amino acids together. It's called a peptide bond, but frankly, it is a bond that we've already seen in organic chemistry that puts those two guys together. This, by the way, is sometimes how you see amino acids written when they are a bunch of them put together in a peptide chain. You'll see the three-letter symbol for each amino acid with a little dash between it that shows that those two are connected to each other. That's how you would name it, which you do not need to do in this class, but it will form this peptide bond when two amino acids come together. So let's talk a little about how we form that peptide bond, and it should look fairly familiar to you. This is essentially the amide bond. So this is the bond we make when we make an amide bond. And just like when we do, we're going to take out water to do so. So in this case, we're going to take out a couple of hydrogens and perhaps an oxygen there. And that's going to come out as water. And basically, when you do that, you're left with just one hydrogen there, no oxygen there, and we're just going to put everybody together. And that's going to make our amide bond that we talked about there in our chapter 14 organic ness that's happening. And that is our carbon oxygen double bond to our nitrogen with our hydrogen in this case. So this is the exact same bond that we saw in organic chemistry in chapter 14, very last thing that we talked about. And that's how basically all those amino acids come together, take water out, and then basically they come together through that connection. As you can see here, basically through the carboxylic acid side of one, and kind of the amino side or the amine side of the other coming together. So here, if we were going to put these two amino acids together, which again is symbolized by that is the order at which we're going to kind of stick them together. Once again here, we're going to kind of bridge through these two guys. We're going to take out water. So again, we're going to remove water. And nothing else will change. Just like in organic chemistry, pretty much nothing else changes. So the entire rest of the amino acid is going to look the same. It's just going to have now that connection point, which is always this what is referred to obviously as a peptide bond here between those two. And once again, on the backside here, nothing much is going to change on this guy as well. And then we'll just draw whatever we got left there, HCOH and CH3. And we got our connection that happens here. And again, we would be our SIR dash THR. So again, although you're not responsible for knowing the names of every one, if you were provided maybe the structures of each of these and you needed to put them together, you might have to draw the peptide bond formation between these two things. So once again, same bond as we talked about previously, just take out the water. All right. Now, naming the dipeptides, nobody wants to name them at this point, right? That's for sure. All right, so we're not naming them either. Don't worry about it. That's all our names. It's a good lecture. It's really just moving, I feel like here. So if we take a look at this, this is just to look at it here. This is a bunch of obviously amino acids together. And once again, you hopefully should be able to recognize our peptide bond that's happening between each of these here. And again, you would split them back apart if you needed to. Right there, obviously, and everybody goes left and right. And basically just builds back to two groups that you need, the carboxylic acid on one without the hydrogen, obviously, and the NH3 group on the other guy there with the plus. And there's our breakage there between those guys. All right, talk a little bit about protein structure. There's primary and secondary levels of protein structure. So the primary structure of a protein is really that amino acid sequence, and depending on sort of that sequence of amino acids that are present, it will code, for example, different types of proteins. Those guys, as we just talked about, is basically held together by the peptide bonds between the different amino acids. And that is what is known as primary structure of a protein, which you do need to know, held together by peptide bonding. And that's the sequence of amino acids that occur. And as you can again see here, everybody peptide bonding here all the way. So we can have just a slight change even at a couple of spots in an amino acid chain, and you will get some different sort of proteins sequenced. So you can see here between these two guys here, the main difference is they got a couple different amino acids happening along the way here at those two spots. And because of that different sequence, again, we're going to start to see some different proteins being coded because of that. Now, we do see some other bonding that's happening here, but once again, all these are going to be our peptide bonds that are happening between our different amino acids. Same thing with insulin here. There's a primary structure that two polypeptide chains. We also see some disulfide bonds happening, and that's the same bond that we also saw in organic chemistry. That is the bond that happens between two thiol groups, right? The SH group. And that happens with cysteine. Cysteine has the SH group on it, the amino acid. So when two cysteine amino acids get near each other, right there is our dipeptide bond, our bridge, disulfide bond, I'm sorry, disulfide bond, our disulfide bridge. And that is absolutely the same bond that we talked about in organic chemistry. Basically, these two thiols coming together. Here we're going to remove H2, right? And go through a oxidation, and that is going to allow those two basically to bond together. So the exact same sort of bond that happened there in organic chemistry. And you can see down here as well. They may not even have to be directly across from each other, but just in the vicinity of each other for those two guys to want to come together and kind of make that disulfide bridge. Let's talk a little bit about secondary structures of amino acids. So secondary structures of amino acids include things like alpha helixes. So the secondary structure is sort of the three-dimensional arrangement of when we get a bunch of obviously polypeptide chains. Polypeptide chains are made up of obviously just a bunch of amino acids hooked together. And you'll have multiple polypeptide chains like we just saw there with insulin, a couple of different chains that could come together. One of the secondary structures is known as alpha helix, and that is a coiled shape that's held in place by hydrogen bonding that is happening between the amide group and the carboxylic acid or carbonyl group of the acid part. And that is happening down the road a little bit. So what ends up happening, because they are not really near each other, they're sort of down the road from each other as they sort of do that hydrogen bonding. That is what causes sort of that coiling shape that occurs with an alpha helix, because they're not like right across the street from each other. They're coming down every four or so down the road, and it's going to cause the whole sort of structure to start to coil with it and make that sort of what's known as an alpha helix. So it's the hydrogen bonding between the hydrogen in the NH group and the oxygen in the carbonyl group. Remember that this is still just like in organic chemistry, a carbonyl carbon, right? Which is more positive here, more negative here. So when we get a NH, which can hydrogen bond, because once again, that is a nitrogen or hydrogen attached directly to a nitrogen. So it's going to be able to hydrogen bond. We will get that attraction and hydrogen bonding that's happening between the hydrogen on the nitrogen and the oxygen there on the carbonyl group. And again, because that is like four amino acids down the chain, that's what's going to cause it to kind of go into that helical sort of shape that we associate, or coil type of shape that we associate with an alpha helix. And that's what we see here. Again, we got our hydrogen bonding happening between the oxygen and the hydrogen down the road. And again, obviously, alpha helicing there, our structure, like DNA, right? Alpha helical type of structure that occurs there. So we do have that hydrogen bonding that is definitely happening in that sort of situation, causing what is again was referred to sometimes as the secondary structure here. Another secondary structure of a protein is what is sometimes referred to as a beta pleated sheet. And a beta pleated sheet is like if you took a piece of paper and kind of folded little squares of it and then unfolded it. This happens actually when polypeptide chains arrange themselves kind of side by side. So unlike in an alpha helix type of bonding that's happening down the road from each other that causes it to kind of coil up, these guys actually are lining themselves up sort of like side by side to each other. So they could kind of just hydrogen bond straight at each other, straight across. The result of that is it gives this sort of pleated sheet type of arrangement, and is typical in fibrous proteins such as silk. And we can kind of take a look at the structure here. So again, the lining up here is a little bit more straight onto each other, the chains. And because of that, they could just go straight across. This is still your carbon oxygen double bond that's happening here. Again, more positive, more negative. And this is still your, if it writes, your nitrogen and your hydrogen happening here. And again, that is the hydrogen bonding between the oxygen and the hydrogen. And obviously the hydrogen and the oxygen is happening here. So again, like if you folded up a paper, put it into a little envelope and you took it out and unfolded it, it's got all the pleats in it. So that causes a different sort of secondary structure that occurs as opposed to when those guys sort of hydrogen bond down the road from each other and kind of coil up on each other. A third type of secondary structure is one that is known as a triple helix. And in a triple helix, it consists of three alpha helices that are basically woven together. Contains large amounts of glycine, proline, in it, which means there's a lot of OH groups basically available to hydrogen bond with each other. And this is what is found in collagen and connective tissue in your skin and cartilage. So this is basically three sort of alpha helices coming together and hydrogen bonding in this kind of triple helix type of form. All right, so take a moment for each of. Okay, so we got side by side hydrogen bonding. That should be our beta pleated sheets. We have our sequence of amino acids in the polypeptide chain. That should be our primary structure. We have a corkscrew shape with hydrogen bonding between amino acids. That's going to be our alpha helix. And lastly, here we have our three peptide chains, which would be our triple helix in this case. Yeah. Any questions on that there? I agree.